These protocols that we develop are important to study the secretion mechanisms in cyanobacteria and their released products, namely the extracellular carbohydrate polymers and proteins. The knowledge generated will allow us to customize these products for several applications, namely biotechnological and biomedical applications. These protocols are very straightforward. They can be tailored according to the producing organism, the user needs, and the final application of the product.
The main advantage of these protocols is that they can easily be adapted to other living systems, in particular to other bacteria. These protocols are easy, however, one may need to introduce some changes depending on the bacterial strain or the degree of purity required.
To begin, cultivate the cyanobacterial strain under standard conditions. Measure growth using standard protocols. Next, transfer the culture into dialysis membranes and dialyze against a minimum of 10 volumes of deionized water for 24 hours with continuous stirring. Centrifuge the culture at 15,000 times G at 4 degrees Celsius for 15 minutes. Transfer the supernatant to a new vial and discard the pellet. Centrifuge again at 20,000 times G at 4 degrees Celsius for 15 minutes to remove contaminants such as cell wall debris or lipopolysaccharides. After the centrifugation, transfer the supernatant to a glass beaker and discard the pellet. To precipitate the polymer,